Thank you guys for the invitation. Thank everybody for coming. This is supposed to be a very interactive conversation about what we do in the lab. We are very interested in a very specific aspect of breast cancer, which is prevention. We understand that there's a lot of amazing scientific groups that are focusing in treatment, and we applaud them and we cheer for them. But we also believe that focusing on prevention, it's something that has to be thought. Diseases are ablated from prevention, and we hope that just one day we can think of that for breast cancer as well. So let's just talk a little bit about breasts. My kids are in the back, so I'm not gonna hurt them or traumatize them much more than I have it already. So breasts are sisters, not twins. So as we develop, we are asymmetric. Even though we have 2 ears, 2 eyes, 2 breasts, they do not develop quite at the same time. So it's not uncommon for you to have breasts that are slightly different in size. And that's why the Victoria's Secret sellers person, sometimes they have a very hard time figuring out what is the one that fits you the best. It's totally normal. It happens to all of us. We think that there's that little say that says that sometimes you carry secrets to the grave. Well, not if you have breast implants. So apparently, a lot of families and relatives discovered that they loved one woman that has been cremated actually had breast implants all along, and it's because during cremation, implants do not get dissolved. So just to say again, some secrets you do not carry to the grave. Smoking can cause breast cancer to drop, but breastfeed doesn't. So breastfeeding does not make your breast look sadder, but smoke does. And I'm not here to criticize anyone for enjoying an occasional cigarette here and there, as one could say that I'm faulted for. But what I'm saying is that maybe you go back to the Secret Victoria's Secret and really finds the little pumps that brings everything up. And if you are as old as I am, you will get the little cartoon. And if you don't, I apologize. But that's droopy, just for context. Nonetheless, breast milk saves lives. So I'm all pro-choice for women to decide to do what they want with their bodies and, use all information that is there to make a decision about how they wanted to live their lives. And I am so glad that we have an ability nowadays that women can choose if they want it or not to breastfeed. I will never criticize anybody for that. I breastfed both of my kids, and I'm very positive about that experience. But in some conditions, breast milk is essential. And I'm talking specifically for those babies that are born very premature. Breast milk brings the first line of immune defense that your kids are gonna be exposed to. It brings the good stuff, bacteria that will really populate their stomach. So then it can help with gases and even colics. So breast milk does save life. And in spirit of October, Breast Cancer Awareness Month, I could not stand here in front of you without talking about screening guidelines, right? All of us, including men, because men does develop breast cancer at a much lesser rate, but they do too. So self-examination, it's very important. Being your advocate when you go to your doctor and say, I have a family history, I have lumpy breasts, I have density breasts. That is the kind of conversation that we have to have with our doctors. If they are not saying you should be starting getting screening before the age that you're recommended to have. We are our advocates. We know our body better than everybody else. And we have the power to have all of those tools in our favor for early detection of breast cancer. And this is very important because, unfortunately, the risk of breast cancer is quite high. 1 in 8 women will develop breast cancer in their lifetime. For those women that develop breast cancer, we know that they have known risk factors only for 30 to 40%. So the majority of women that are developing breast cancers, they do not have a family history. They do not have alterations to the genes that increase the risk to develop breast cancer. So surveillance is very important. Education is very important. And the research that we are doing here, as well as other scientists at Cold Spring Harbor that focus on breast cancer, it's really important as well. And we like to think about pregnancy being one of the risk modifiers of breast cancer. So every time that I talk about pregnancy, we think about a bump that grows, and then the baby that is developed, so much love, it's born, and then nights of sleep, not sleeping for many years, maybe decades. But what we know now is that in addition to the baby developing, the bump growing, and the baby being born, there are many things that happen during pregnancy. Our whole body is exposed to alterations because of the pregnancy hormones. It really changes how our body works. For example, we as women that are pregnant, we have an increased cardiovascular system, and that has the main reason of increase the blood circulation to accommodate a growing body that is then serving as the housing for the developing baby. We have a slightly altered immune system. Again, this is for 2 reasons. The first reason is to make sure that the mom's body is not going to attack the baby that is developing, but also then in preparation for lactation to bring in all the memory of viruses and bacteria that we went through and produce that in the form of antibody that then is given to the baby, again, as a form of the first line of defense, immunity defense that the baby will develop. Some may say that our brain works differently. I like to call that the mommy brain. It starts very early when you're pregnant, and then you kind of wake up 5 times before your child wakes up hungry. It's that connection. You're very focused on the baby's well-being. Everything else, it's kind of like background noise. So there are changes in how your brain works because of the pregnancy hormones. And all of those changes have been placed for many, many years through our evolution to make sure that the baby is there to having all the nourishing and attention that it's needed. And we focus on how breasts are changing after pregnancy. So all of us in the audience here, men and women, we have some sort of breast tissue. Our breast tissues developed when we were in our mommy's belly. The pregnancy hormones that our mommy's belly had induced the growth of the breast tissue for men and women. The biggest difference between men and women here, it's because during puberty, women experience a higher level of estrogen, which really allows the breasts to further develop. So we all here have breast tissue. And until pregnancy comes into place, breasts are not really functioning. Breast's main function is to produce milk to support the offspring. And because of that function, pregnancy really changed how breasts look from a tissue perspective. So here I'm showing to you something that we look a lot at the lab, which are pathological images of a breast from somebody that is not pregnant. So all those little purple dots here, those are breast cells. The little white spot right in the middle of those cells is where the milk gets dropped and then gets delivered to the baby. During pregnancy, gestation, those cells start expanding. Those structures start growing because they collect the signals that says there's a baby coming. We have to multiply so that we can have enough milk for that baby. And this is what happens during lactation. Look how filled this tissue is with all those pink, purple-like structures. All of those structures right here are now secreting milk into the middle of those, this white spot here that will go to the baby. And that can last until lactation goes. So some of us decide to, it depends on how long some of us decide to lactate for. At the end of lactation, meaning that when there's no suckling, when the baby stops nursing, the breast undergoes a process that is called involution. That means that all the cells in the breast that were once making milk, now they call the immune system to get them out. Like we've done our job. We don't have to be here anymore. And the immune system really clears them out to the point that when we look at the tissue, it looks like the tissue of somebody that has never been pregnant. It's that dramatic change. And this is all in response to the hormones that are during pregnancy and lactation. So pregnancy itself has a very important function on developing of the breast. And how we like to study that? We like to study that from an epigenetic perspective. So this is a big word to say that you have your DNA inside of the cells, and there are products of your DNA that is made inside of your cells. Some of the products are called RNA, and I'm going to be referring to them as the ID tag that we use. So every cell has a combination of RNAs that then when we look at them, we can tell which cell they are. If they are a muscle cell, a heart cell, a cell of the breast that makes milk, for example. That's what gives their identity. That RNA gets them produced into protein, which I will be referring to as a profession. The protein is the readout for what the cells do as a function. So when we look at the proteins that we have in the breast, that's their function. So the muscle cells will contract, the breast milk will secrete milk proteins. In the way that we study those developments, we like to think of an on and off system. Every time that I say that DNA part is off, it means that the ID tag, then it stopped being produced, and you do not have a cellular profession that defines what the cells has to do. Every time that I say that the part of DNA is on, it means that you have increased production of the genes that define that cell as an identity, and a lot of the proteins that define that function. And I'm gonna get to why I spend so much time explaining that, because we believe that is understanding the switches of on and off of breast cells. It allows us to think of how milk production genes get turned on in response to pregnancy hormones, but how we can then shut down genes that are associated with cancer development. So how do we study that? We are a modeling system lab. So we model disease and we model develop in rodents and in cultured cells. So how do we do that in vivo models? So we have the ability to introduce foreign DNA to the cells of a mouse, for example, a piece of DNA that has been associated with increased risk of breast cancer. And now we have that animal have the genetics of a woman that has increase of breast cancer risk. So now we can study how to modify their risk. What is a component to their risk? How we can then prevent those animals to develop breast cancer. We have also been using ex vivo models, and this is something that we are super excited about because it allows us to move our science from animal models into tissue that women donated to the lab. We call those organoids, and what they are, so we received tissue fragments that are donated by women that are undergoing cosmetic plastic surgery in collaboration with Northwell Hospital System. We kind of chop them in little pieces and we put them to grow in a three-dimensional dome. By allowing them to go in a three-dimensional setting, we are kind of reproducing a lot of the cell-to-cell contact that we will have in our breast. So cells behave very similar in those cultures as they do in a human breast. How do we know that? We can grow those cultures with pregnancy hormones and we can make those cells make milk. So we can now study a lot of the biology that we have been relying on animal models into study the biology in tissue that is donated by women. And we are very excited about that, and I will go give you examples of why. So When it comes to prevention, we have to have a model system that is predisposed to develop breast cancer. And the one that we chose to start our program with was the BRCA gene. Everyone in this room here has the BRCA1 gene, but people that have mutations on the DNA that is part of the BRCA1 gene have an increased risk to develop breast cancer. What is puzzling, though, is that it increases the mutated BRCA gene, it increases substantially the risk of breast cancer, but that does not mean that every woman that has a BRCA1 mutation will develop cancer. So we are very interested to understand what is happening to the 35% of the cases that do not develop breast cancer. Can we understand the biology of the normal breast? So then we can predict who will develop the cancer, who has the gene, mutated gene, and prevent cancer. So using the example that I gave before, we develop a mouse model that then we inserted, that we remove or altered the BRCA gene of the animals. It's a very cool system because we can really track tumor development in those animals. This is the breast tissue of a mouse at 12 weeks. Looking, and here is just resembling, representing initial cancer lesions. There's a lot of growth in here. A lot of those empty spots, the pink spots are being covered with the purple spots. And this is an advanced cancer lesion with the majority of the tissue now is purple. So we can study a lot of the progression of the cancer in those animals. We asked the question, would pregnancy change the risk of breast cancer here? Why? Because women that are pregnant before the age of 25 years old, they have a 30 to 40% decrease on their risk to develop breast cancer. This is known for many, many decades. We still don't understand why a fraction of women that are pregnant at early age have this preventive anti-cancer benefit of pregnancy. And in those animals, for example, when we put them through pregnancy, so this is a curve that it shows that the amount of animals that develop cancer. Every time that you have a drop here, it means that a bunch of animals got cancer. So the animals of the nulliparous, which is another word for never pregnant mice, they drop very fast, meaning that all the animals in that cohort develop cancer. The pink line shows animals that have been put through pregnancy. And you can, there's a few that do, but the majority of them don't. And now we have been using a lot of the techniques that we have been developed over the years, which is to study the cells and to study the DNA of the cells, combining cellular analysis with computational analysis to understand why those tissues here remain cancer-free comparing to those ones that then develop into cancer. So we found a few things that I wanted to cover today. So we remember that I've told you that there are the parts of the gene that got on and off. One of the things that we found that gets turned on after pregnancy is this event of senescence. Senescence is pretty much what we are observing outside right now, is this ability to grow old. This is why the leaves on the trees, they change color in autumn. So they are not growing anymore. So they are turning yellow because they are really turning down all the programs on them, and eventually they're going to fall off the trees. So we learned that cells after pregnancy turn on that system, and that's perhaps why they cannot turn into cancer. Unlikely trees that the leaves just shed out of the tree. What we know is that when there is a senescent cell inside of our body, this is one of the ways that the immune system comes and says, something is not right here, I'm just gonna get this cell out. And that takes us, look, we spent so many years studying breast cells and so many years building this model, and now the model is telling us, you're not quite looking at the right cells. You have to look at other cells of the breast that are there and maybe having an impact on how cancer development. So it took us to look at the immune cells of the breast. But the complication here is that there's so many immune cells. How do you even start? How do you even pick? So we use an approach in the lab that is called single cell. So the approach says it all. The name says it all. You get a piece of a tissue, you chop it out in the point that you have cells just floating in the liquid, and you put that through a machine that will sequence the ID. Remember the product of the DNA, the RNA in each one of those cells. And by looking at the RNA, the ID tag of those cells, we now can figure it out which cell is more important to prevent cancer development, which we did. So we found, so this is, we call this blood flow cytometry. This is pretty much how, when you go to get your blood test done, this is pretty much how the doctors, they say, they see that you have different kinds of red blood cells and white blood cells in your body. It uses cells, size, and cell markers to identify a cell lineage. So we show that there's more of those cells in animals that do not develop cancer. And because we can engineer animals to introduce perturbations, if we now use a mouse model that even after pregnancy, those cells are not going to be expanded, meaning in a lot of abundancy, now we see lesions of tumors even after pregnancy. Really making a direct link with the presence of the cells in the breast and the appearance of cancer. Why we are so excited about that? It's very hard to target epithelial cells. So we have to talk about perhaps gene therapy here. But immune cells, for a very long time, we have been pretty good about waking them up when we had to, and with vaccination. So we were just witness of a huge vaccination enterprise that actually allows many people from dying because of COVID. And I think that this is one of the proudest moments that we have in science, to the point that the Nobel Prize acknowledged that importance of the discovery for humanity. And because now we have found that immune cells have this role on preventing cancer, we can now understand what kind of antibodies those immune cells are secreting, synthesize them in vitro, make them in a vaccine formulation, and give it to animals, which I'm happy to say that we just started doing this week. So I talked to you about pregnancy as a risk modifier of cancer. And I think that that was what we needed to develop the models and develop the hypothesis. As scientists, we chase hypotheses to understand cancer risk. And the beauty of science is that every time that you close a door, hundreds of other doors open, because it allows us to understand complex systems better. And now we understand that there are other types of life experiences that woman goes through that change the risk of breast cancer. And we have projects now understanding the effect of aging on breast cells and how that increases the risk of cancer, menopause, UTI, some sort of syndromes, or other types of inflammations that women experience throughout life. Sometimes the risk of developing those inflammations are at the same risk that one has to develop breast cancer. And we are now trying to connect the dots between their presence and the risk of cancer and how we can then prevent cancer. And I'm happy to address any questions that you have about this slide here during the discussion session. I stood here for perhaps way too long, but the people that do the work that I'm here talking about are those beautiful people here. I would say that most of them are here. So here we have an array of different projects that we have in the lab that I'm also happy to address. We work hard and we play harder. So I'm extremely lucky and glad to have such an amazing team to work with. And in addition to that, I have lots of people at the lab that help us to move forward what we do. We also interact with breast cancer advocates. We were just there yesterday at a Cancer Center breast cancer survival supporting group, talking to women that have the diagnosis of cancer, that have been treated with cancer, and that really changed the perspective to how we do our science. We do our science because we are curious, because we are in a place to ask risk questions, but we also wanted to do science that benefit for the benefit of the patients. And thank you for your attention, and I'm happy to address questions. Thank you very much.